Benvenuti.
Welcome to the course on Physiological Regulating Medicine. It is a great pleasure and honor for us to share our knowledge of physiological regulating medicine with you, to bring your attention to the results of our research, and to share the possibilities for application of this extraordinary modern medicine. We will follow a challenging path that will be a journey within physiological regulating medicine. In these first stages, we will focus our attention on the foundations of PRM, and we will try to understand the working mechanism of the pharmaceuticals of physiological regulating medicine. Only by knowing these aspects will we understand how to impose a correct treatment according to the principles of physiological regulating medicine, and this will be the objective of our journey. We normally like to talk about the treatment triangle. This triangle is composed of three parts. A blue part, the base, is drainage, which we'll discuss at length. The yellow part is definitely the most innovative. We will discuss psychoneuroendocrine immunology, and we will understand how to balance the so-called PNEI axis. Then there is a third side of this triangle, the red side, and it is the simplest side. Or rather, we will understand that to treat a patient, we must not only detoxify and drain him and restore equilibrium to his PNEI axis, but we must also alleviate his symptoms from the beginning. However, and I want to emphasize this concept right now, PRM, physiological regulating medicine, is not only aimed at modulating a symptom, but is directed at achieving a profound healing of the patient. For this, we will dedicate considerable attention in these phases to the blue part, that is the drainage, and to the yellow part, that is the rebalancing of the PNEI axis. Because this is a true decisive key to curing the patient instead of just modulating his symptoms. So, if we are ready, we can begin this amazing journey into physiological regulating medicine. You will see that physiological regulating medicine can certainly be considered to be the most modern integration between conventional medicine on one hand and non-conventional medicine on the other. An evocative image may allow us to better understand this concept. PRM is a bridge that connects homeopathic tradition with state-of-the-art molecular biology. We will also take a look at pharmaceuticals that are the most important expression of this medical approach, and they represent none other than a bridge joining animal, plant, and mineral derivatives that are the typical players in homeopathic pharmacology. Together with neuropeptides, interleukins, hormones, and growth factors that are the typical players in state-of-the-art molecular biology. There are many roots that form the basis of physiological regulating medicine, which derive certainly from homeopathy, and a big thanks goes to its founding father, Christian Samuel Friedrich Hahnemann. Without Hahnemann, neither we nor you would be here to talk about physiological regulating medicine. We also give great thanks to Dr. Hans Heinrich Reckeweg, the founder of homotoxicology. Homotoxicology represents the most modern evolution of classic homeopathy. Many players of Dr. Hans Heinrich Reckeweg's pharmacology can be found in the pharmaceuticals of physiological regulating medicine. However, if we only represented our historical roots of homeopathy and homotoxicology, you might ask, what's new in PRM? The truly big news is in an acronym. P, psycho, N, neuro, E, endocrine, I, immunology. It's no coincidence that I have written the letters vertically, because I want to convey the holistic sense of this acronym. We are a network, a neuro-immuno-endocrine network, wherein three major systems of homeostatic control, the central nervous system, The endocrine system and the immune system are closely interconnected with each other. Every change that occurs in our body is the expression of a comprehensive variation in the psycho-neuro-endocrine immunological axis. An example may clarify this concept in a very simple way. We all know the psychosomatic direction of diseases, from stress to gastritis, or from stress to cancer. This is the classic expression of a PNEI imbalance. We should also rethink another direction in physiological regulating medicine, the somatopsychic direction. For example, during the perimenopausal period, a classic instability in mood is frequently observed. If we limited ourselves to distinctly viewing the three major systems, we could say that this instability is due to low serotonin levels. Certainly, this is the effect, but the low serotonin level is linked to an alteration in the incretion of the female sex hormones. So to remove the cause of this hypothetical perimenopausal depression, we should not only act on the low serotonin levels, but also on the basis where the real disturbance originated. This is a methodological pillar of physiological regulating medicine. We need to be able to properly consider the body, and that's what we'll do together, like a network. And the key word is neuro-immuno-endocrine network. Every alteration will be an alteration in the players of this neuro-immuno-endocrine network, the so-called messenger molecules, that is, the neuropeptides of the central nervous system, the hormones of the endocrine system, and the interleukins of the immune system. To understand this in a very simple way, we consider these messenger molecules as the words and messages produced by these three major systems. They are the words with which the nervous system communicates with the endocrine system. These words are the neuropeptides, and the hormones are the messengers with which our glands inform our body about what is happening. One of the keys of PRM, physiological regulating medicine, is that every pathology is the expression of an alteration in the concentration and working mechanism of these molecules. What is very interesting is that when this network is in a state of homeostatic or a physiological equilibrium, the players, neuropeptides, hormones, interleukins, are in a particular concentration. As we know, this concentration is a physiological concentration. Only if these substances are present in the extracellular matrix, the so-called biological milieu, in the right quantity can we be referred to as being in a physiological condition. When the concentrations of these substances become hyper or hypoconcentrated, we are in a pathological state. After years of study, today, we finally know the physiological concentration in the extracellular matrix, and you will discover something that is extraordinarily amazing, which is that the physiological concentration of the messenger molecules in the extracellular matrix is a homeopathic concentration. Our body works at homeopathic concentrations, and we have reproduced these concentrations. We said that we are a neuro-immuno-endocrine network governed by fine control mechanisms, and by fine control we mean precise ligand concentrations, where the ligand is the messenger molecule, either a neuropeptide, hormone, or interleukin, that binds to its transmembrane receptor. Today, after many years of research, we know the physiological concentration of these messenger molecules in the extracellular matrix. What is extraordinary is that the method of preparation of the homeopathic pharmacopoeia, dilution and dynamization, or as we prefer to call dynamization today, sequential kinetic activation, makes these messenger molecules even more effective. The gift that Dr. Christian Friedrich Hahnemann left us is truly extraordinary. Therefore, in our research, we have begun to test in vitro and in vivo the effectiveness of these messenger molecules. We have also tried to use different concentrations. For example, at first we tried those between the mother tincture and the so-called 10 to the power of -3 dilution. For clarification, in terms of weighted concentrations, we're referring to milligrams per milliliter. You can understand that these molecules, particularly the interleukins, which are very potent, have only toxic effects at this concentration, and is therefore not possible to use them for a pharmacological purpose. Our research was directed at the search for new molecules in pharmacology. Therefore, between 0, mother tincture, and 10 to the power of -3, it is not possible to use these substances. We could try to go lower. In fact, we have not tried to go lower ourselves, but the traditional pharmaceutical industry has tried using these substances, particularly the interleukins, at a concentration between 10 to the power of -3 and 10 to the power of -6. For clarification, in terms of concentrations, we are referring to micrograms per milliliter. We can define this concentration as a pharmacological concentration with extraordinary therapeutic effects, but also with severe side effects. It is therefore not possible to use such concentrated substances at the level of micrograms per milliliter. This is where we will have inserted ourselves. We have tried something revolutionary and tried to create a utopia. If we go under the minimal effective pharmacological dose, 10 to the power of -6, what will happen? We have seen something very important when we tried to use these substances in a range of concentration between 10 to the power of -6 and 10 to the power of -15. These numbers might not tell you much, so I prefer to explain the concentrations. We are referring to nanograms, to picograms per milliliter. It is an amount that seemingly corresponds to almost nothing. But if we do something, this almost nothing becomes as effective as the pharmacological concentration. What is this new element that we are introducing? We have observed one thing. If we don't shake according to the method of homeopathic preparation, referred to as dynamization, which is now being called sequential kinetic activation, these low doses and low concentrations, in the order of nanograms, picograms, and femtograms, do not have any therapeutic effect. But if that low dose corresponding to the aforementioned physiological concentration of the extracellular matrix is subjected to the homeopathic preparation method of sequential kinetic activation, that low dose becomes as effective as the pharmacological concentration of micrograms per milliliter, with the great advantage of no observed negative side effects. Can it be true? Please, do not just take my word for it, but believe only in the experimental results. We have carried out this experiment not as the Guna Scientific Department, but in collaboration with the Institute of Human Anatomy at the University of Milan. We have conducted 2 experiments, one in vitro on cells and the other in vivo on mice. This is important because you know that when you work with such extremely diluted microdoses, there might be a placebo effect. Well, I ask you if it's possible to influence a fibroblast with thought, or the placebo effect, or if it is possible for me to convince an allergic mouse that he is healed thanks to the placebo effect. We took some human fibroblasts and subjected them to the action of different concentrations of the interleukin called interleukin-1. Interleukin-1 is very interesting because it is a pro-inflammatory interleukin and a messenger. Remember when we said that these messenger molecules are the words with which our control systems speak? Well, interleukin-1 is a messenger of the immune system, And the message that this interleukin carries to other cells in the body is, guys, inflame yourselves. We know that biologically, inflammation is the body's appropriate defense response. But to say inflammation, or to say increase of function, as you well know, means the same thing. We therefore put these fibroblasts in contact with interleukin-1. Interleukin-1 immediately bound to the transmembrane receptor. After binding, within the fibroblast, the end of the world occurred. A flood of chemical reactions, various substances were produced, and there was one in particular that interested us. This substance is called NF-κB. It is a substance that all of us have in our cells and is a nuclear transcription factor. What does this mean? This substance, NF-κB, produced by the cell after the binding of interleukin-1, the inflammation messenger, and the transmembrane receptor, is capable of entering within the nucleus and activating some parts of the DNA, so the cell produces other substances. Well, according to the International Experiment Criteria, measuring the quantity of NF-κB in a cell means measuring the effects of the ligand on the receptor. The results were surprising, especially for us. Let's look at them together. The cell was bound with various concentrations of interleukin-1. We have a first result for the positive control. For example, when incubating the cell with a lipopolysaccharide. And this is the quantity of NF-κB that it produces. We see the quantity of NF-κB that the cell produces if put in contact with a placebo, for example, water and alcohol. This is result A. Now we see what happens by incubating the cell, the fibroblast, with a pharmacological concentration, for example, micrograms per milliliter of interleukin-1. This is the concentration that we have seen to have extraordinary therapeutic effects, but also, unfortunately, severe side effects. This is result B. As you can see, the amount of NF-κB produced by the cell in this case is very high. What is extraordinary, and this is the big result, is that the quantity of NF-κB produced by the cell following binding with low-dose physiological concentration, that is picograms, femtograms of interleukin-1, is result C, which is practically the same as that produced by the cell incubated with the pharmacological concentration. However, in using this concentration, which will be the concentration that we will use in our pharmaceuticals, there are no observed side effects for the cell. But that's not all. If we incubate the cell with the very low concentration that we normally use, for example, picograms per milliliter, without it having been agitated or dynamized, or if you prefer to say, kinetically activated, as we can note, this is result D. Well, that low dose is absolutely ineffective. Only a physiological low dose subjected for the pharmacological and pharmaceutical preparation of sequential kinetic activation has and demonstrates the same effects as an allopathic pharmacological concentration, but without the side effects. You can understand that this opens an enormous, extraordinary world of therapeutic possibilities. We all feel now that we have an exceptional therapeutic weapon in our hands to cure patients and restore their physiology without any side effects. Another line of our basic research was directed toward in vivo research on mice. We have measured the effects of some interleukins at different concentrations in the treatment of asthma. But before seeing the truly surprising results of our experiments, we have to take a little detour to discuss how the immune system functions. We could talk for hours, but we must focus our attention on what is fundamental. To fully understand the role that the immune system plays in determining a disease, imagine the immune system as a scale consisting of two plates. We will call one of these plates Th1 and the other Th2. Th refers to T helper lymphocytes 1 and 2. Every disease in which an immune etiology is recognized as the expression of an imbalance of this immune scale. Certain diseases have a predominance of the function of Th2 lymphocytes, e.g. allergies, asthma, and atopy, and other diseases are the result of a hyperexpression of Th1 lymphocytes, e.g. psoriasis, Crohn's disease, or rheumatoid arthritis. It is intuitive that in order to profoundly cure, and this is our objective, a pathology that is based on an immune imbalance, our objective will be that of restoring the balance day after day, and, I would say, month after month. How can we do this? We can do this by using these substances that we have defined as messengers of the various major homeostatic regulation systems that are, in the case of the immune system, the interleukins. Looking at this figure can help us to understand how to use these substances and to understand what happens when a patient has asthma or an allergy in general. We have some lymphocytes that we call Th0 lymphocytes. To illustrate what Th0 lymphocytes are, we can imagine them as lymphocytes that have not yet understood what they want to do when they grow up. For example, if they want to become Th1 lymphocytes or Th2 lymphocytes. If they become Th2, they may be responsible for allergy. If they sway too much towards the Th1 side, they can be responsible for autoimmune diseases. In allergies, we have an absolute predominance of the Th2 side. This Th2 side produces some substances, the messengers of the immune system, which are the interleukins called interleukin-4 and interleukin-5. They are the real cause of allergies, that is, their excess is the real cause of allergies. I, for example, am not allergic since in my extracellular matrix these interleukins are within that concentration range that we know precisely to be between 10 to the power of minus 6 and 10 to the power of minus 15. In an allergic subject, these substances are in excess, and we can say that they are in up concentration. Why are they responsible for the allergic reaction? Because, remember, interleukin-4 is capable of sensitizing the B lymphocytes, and the B lymphocytes affected by this messenger react by producing antibodies. These antibodies are IgE antibodies. The IgE antibodies do their job, which is to adhere to cells called mast cells, sensitize them, and upon a second contact with the allergen, make them produce a substance called histamine, which is what is truly responsible for allergic symptoms. Remember, at the beginning we said that we want to treat the person and not only his disease. That is, we want to remove the cause and not act only on the effects. Well, in the medical world, to cure allergies, we can act only by modulating the production of histamine. That is, by trying to control the symptoms. Through physiological regulating medicine, we'll work on the Th1 to Th2 scale imbalance. In what way? Let's try to enter into the immune system. Let's try to take a journey within it. We have understood that we have Th1 and Th2 lymphocytes. The Th1 lymphocytes produce different interleukins, of which two are particularly important and should be remembered. Interleukin-12 and interferon gamma. The Th2 lymphocytes produce various interleukins, and if you only remember one of them, please remember interleukin-4. Why are these three interleukins so important? We could call them the fundamental interleukins of the immune system, since they are capable of influencing the development of the Th1 or Th2 lymphocytes. Let's see how interleukin-12 induces what in immunological terms is referred to as the upregulation of Th1 lymphocytes. In other words, it is a messenger that says to the Th1 lymphocytes, "Hey guys, work hard. Interleukin-4 is the one that is capable of inducing the upregulation of the Th2 lymphocytes. It is evident that if the patient has an allergy, and the allergy is an expression of an excess of Th2 lymphocytes, the cause is interleukin-4. But let's see what these interleukins are still up to. The interferon gamma produced by the Th1 lymphocytes is able to do something that is very important to us in terms of therapy. That is, to induce the so-called down-regulation of the Th2 lymphocytes. Understand that interferon gamma is very important in curing the allergy. Interleukin-4 is also able to induce down-regulation, which in this case would be down-regulation of the Th1 lymphocytes. So, if in the case of an allergy, we have an excess of interleukin-4, or actually an excess of Th2 lymphocyte function, which will be the two interleukins able to balance this scale week after week? The response is obvious. Interleukin-12 to stimulate the Th1 lymphocytes, and interferon gamma to downregulate the Th2 lymphocytes. This is what we normally use in therapy. For example, as a very important terrain-related and basic preparation, the only medication in the world that can work not on the effects of the allergy, for example the release of histamine, but on the cause that has led a certain patient to be allergic, for example a Th1-Th2 imbalance, is the preparation called Guna Allergy Prev. You will note that within this preparation, which we will later study in depth, there are present not only homeopathized plants, homeopathized animal derivatives, and homeopathized mineral derivatives, but in particular, two interleukins are present. Which ones? Interleukin-12 and interferon gamma, which determine the stimulation of Th1 lymphocytes through a cross-regulation mechanism, which is good for someone who is allergic, and a modulation of Th2 lymphocytes, which is extraordinary for someone who is allergic. Now that we know how the immune system functions, let's explore our laboratory mice. Let's see what has happened to their immune systems. We have taken a mouse that was in fine condition, and we will call him Lucky Mouse. We began giving him intraperitoneal injections of ovalbumin plus aluminum hydroxide, which is the method to induce allergies in a healthy mouse, in accordance with the regulations of international experimentation. When I say healthy mouse, I mean that our Lucky Mouse has an immune scale that is perfectly balanced. We performed an initial intraperitoneal injection, and after 7 days we repeated the intraperitoneal injection with ovalbumin and aluminum hydroxide. After 13 days, we subjected the mouse to an aerosol treatment with these substances. Well, after 18 days, we can say that the Lucky Mouse is no longer so lucky. He is now an allergic mouse. When I say allergic mouse, I mean that he manifests the typical symptoms of allergy, e.g., bronchospasm or cough. But this has just a partial significance for us, especially after what we have learned about the immune system. When I say allergic mouse, I mean to say that his immune system is unbalanced. In fact, in immunology, we say that he has had a Th1-Th2 switch. If I say that he has a hyperfunction of the Th2 lymphocytes, I mean to say that he is producing too much interleukin-4, and we will find this interleukin-4 in his serum, in addition to in his tissues. Well, this is what we have done. After 18 days of pro-allergic treatment for our mouse, we measured the interleukins in withdrawn blood, and we saw that the interleukin-4 level was extraordinarily high. However, we know how to restore equilibrium to the immune system. In this case, if we have an excess of interleukin-4, as the serologic data show us, we can compensate for the imbalance by using, let's say, the two anti-allergic interleukins, interleukin-12 and interferon gamma. This is what we have done in the mouse, and these are the results. We gave the mouse a pharmacologic concentration which, if you remember, corresponds to micrograms per milliliter of a combination of two anti-allergic interleukins, interleukin 12 and interferon gamma. The results were extraordinary. After 7 days of therapy, the mouse was no longer allergic. That is to say, his immune scale was no longer imbalanced, but was returned to a physiological state. Sure, by using micrograms per milliliter of these two interleukins, we cured the mouse very well. The mouse is no longer allergic. But the problem is that now the mouse is no longer anything, because he's dead. This is the side effect of the very high concentration, micrograms per milliliter, of these two interleukins. We then tried to use our physiological concentration. You may remember that I spoke of picograms per milliliter and femtograms per milliliter. If we speak in acronyms, it will be simpler. We used interleukin 12 at 4Ch, the homeopathic dilution that is on the scale of picograms per milliliter and femtograms per milliliter, and we also used the combination with interferon gamma at the dilution of 4Ch. Remember this dilution because it will be the dilution that we will find again in the preparations of physiological regulating medicine. We administered in the same ways as for the pharmacological concentration the two cytokines. We can now call them guna to the mouse, and these are the results. Exactly the same as the results of the pharmacological concentration. We have reduced the level of interleukin 4, that is to say, we have rebalanced the immune system, but the mouse is not only healed from the allergy, he is not dead. So in that sense, we have not had any side effects. Another piece of data is extraordinarily interesting. By using these interleukins, we have seen that the level of friendly, anti-allergic interleukins, for example interleukin 12 and interferon gamma, are increased in the blood of the mice. What is extraordinary, and what emerged from our experiments, is that if these low-dose physiological interleukins are used only diluted and not kinetically activated, or if you'd prefer to say, dynamized, these interleukins do not function. Only if they are prepared homeopathically through dilution and dynamization do these interleukins have the same extraordinary therapeutic effects as the allopathic pharmacological concentration, but without the side effects. The data on the increase of these two interleukins in the blood of the mice is very interesting because it gives a sense of the biological therapy. I ask you this question: how is it possible that, by administering femtograms, for example an almost inexistent quantity, to a mouse, we find these substances in quantities of picograms in the blood? It's crazy. It's like depositing 100 euros in the bank and then coming back to find we have 10,000. It's a fairy tale. But not a fairy tale in our immune system, because our immune system in this case performed a mechanism called upregulation. For example, these two interleukins have stimulated the cells of the immune system. We know exactly which ones: the dendritic cells to produce their own interleukin 12 and interferon gamma. This is the meaning of biological therapy and of physiological regulating medicine. By using physiological concentrations, the same what we have in our body, we are able to induce, restore, and reacquire the physiological capacity for autoregulation. Another piece of data that I want to bring to your attention is that of the quantity of eosinophils in the bronchoalveolar lavage of the mouse. From the data, we can see how the quantity of eosinophils in the BAL of the allergic mouse is very high. The quantity of eosinophils after treatment with micrograms of allopathic interleukin 12 and interferon gamma, for example treatment with great results but side effects, is therapeutically extraordinary, with 0 eosinophils after a few days. But the mouse is dead. The data is even more extraordinary. By using the physiological concentration 4Ch of interleukin 12 and interferon gamma, the eosinophil count in the BAL is still the same value of 0 as with the pharmacological concentration, but without any side effects. The last figure that I want to bring to your attention regards the safety of these substances, because one of the questions could be the following. Well, if I use interleukin 12 and interferon gamma, I re-equilibrate the immune scale, that is, the Th2 lymphocytes and the Th1 lymphocytes, and day after day this happens and this happens. But it won't be that by continuing the therapy, an unbalancing of the other side of the immune system could occur. That is, I cure the original disease, but I induce an autoimmune disease. Well, the results that I am showing you allow us to understand the absolute safety of the concentrations we have identified. You can note that by using allopathic concentrations, micrograms per milliliter, a high level of interleukin 12 and interferon gamma, with respect to the mouse's physiological level, remains, even after the end of the treatment. This is the meaning of an allopathic therapy. Look at the data on the use of the 4CH, physiological low-dose concentrations of the two interleukins. After an initial spike, the body, with physiological regulating medicine, auto-regulates itself and returns to the physiological blood concentration levels of interleukin 12 and interferon gamma. This exemplifies the absolute certainty of not counter-unbalancing the immune system toward the other side. This has given us a great safety profile, and that is at the basis of the formulation of preparations in physiological regulating medicine.